pyruvate. Just a practical thing that I have just found out, so I haven't been able to do anything and probably will not. We have a new recording system. The recording system is limited to exact times. So therefore, if there is any lecture going on after 50, it will be cut off, and I've just been told you will not be able to get it. We have been able to work out with the faculty that come in and set up. Sometimes people are late, I get started late, etc. But your recordings are limited to 9 to 9:50, and when the lecture goes a minute or two late, you are not able to get them. I have just been told this morning. I am going to try to see what can happen, but I'm told that's the system. If any of you would like to see all of this, it wouldn't hurt to just send an email to the IT. This is especially important to the viewing audience to realize that the last minute or two of lectures are likely to be truncated. I can't do anything about it. Okay, now back to metabolism 1. The fate of pyruvate is where we are in this overall glycolysis. We're going to finish that up today. Now, I had asked some questions. People had been asking about slide 2.6. That equation is net balance. So as it is written, it is correct, because the net, remember, in glycolysis, the net production uses 2 ATP and makes 2 ATP. Because when you actually cross out, you're actually essentially using, you actually are using 2 ATP and making 4. So the net essentially is to make 2. And that equation in slide 2.36 is that net equation when you add everything that happens. Okay. Sometimes when you're in the middle of a lecture, it's like I have a feeling that if you ask me what my middle name is, I have a 50-50 chance of getting it right. It's just a whole different mindset. Okay. So we were on the last slide in pyruvate utilization. We learned about what happened in muscle, and this is the one we're mostly concerned about in this course. The most efficient utilization of pyruvate occurs through aerobic oxidations and respiration. So what actually is involved, you know the enzymatic part of it, the oxidative decarboxylation by pyruvate dehydrogenase complex. This is taking place inside the mitochondria, so that's why the pyruvate that we make from glycolysis has to cross the mitochondrial inner membrane through a transporter facilitated diffusion system. But at this point, we then go through further decarboxylations through the citric acid cycle, because our pyruvate dehydrogenases are linked between glycolysis and TCA cycle. The final oxidation, in other words, we're really trying to do the oxidation of glucose to CO2 and water. After pyruvate dehydrogenase, we have done the oxidation of glucose to acetyl-CoA. After the TCA cycle, we've done the oxidation to reduce nucleotides plus CO2, and it takes respiration, or the respiratory chain, to convert those reduced nucleotides to get that water out. So this is the reaction. It's highly favorable. It has multiple complexes, and you know all about that from Dr. Ponticelli. What we're focusing on here, of course, is what are the cofactors that you need. For instance, if you're missing thymine pyrophosphate from a nutrition point of view, from a metabolism point of view, this isn't the only enzyme that won't work. We have to be careful about our additions of NAD+ and NADH. Since this is inside the mitochondria where we have respiration going on, we don't have as much of a problem doing our regeneration as we do when we have glycolysis out in the cytoplasm, where the major adenine nucleotide NAD+, NADH pool is not located. So as we go through this, we'll be looking at, in the TCA cycle section, we'll go through this in a little bit more detail, focusing on the regulation. So the overall regulation of glycolysis can be looked at here. And here you have, this is typical inhibition up here from feedback inhibition. You learned about these compounds. We don't know yet what fructose 2,6 is actually doing, but to see that along the pathway there are multiple control steps. And I'm focusing on this one, between fructose 6-phosphate and fructose bisphosphate, and that's PFK1. What we'll also be adding to the mix is when our glucose goes to glucose 6-phosphate, and that sort of traps it inside the cell, and now we're going to trap it for glycolysis. There is an interplay between the polymeric form, which is glycogen. And so we'll be looking at glycogen synthesis and breakdown and how these two systems interact. So the major pathways, the regulation, what you should be looking for that you need to understand in this section is glycolysis, the breakdown of glucose, gluconeogenesis, the formation of glucose, glycogen breakdown, and glycogen formation. You need to know those pathways and how they are interrelated. Okay. Now, PFK1. This is a slide that appears later on in the notes, like very close to the end on the PFK2. And I'm going to go over it now to see, hopefully you can follow it, because we've had a portion of it, and it will be familiar to you when we actually have to go over it again. So if you look at the outer sections of this, this represents the major pathways. Fructose 6-phosphate by PFK1 goes to fructose 1,6-bisphosphate. That's the reaction we've talked about. Okay? So we can use the reverse of that when we want to make glucose and go from fructose 1,6-bisphosphate to fructose 6-phosphate, and the enzyme is fructose 1,6-bisphosphate. These outer circles, this R or these outer arrows, represent major pathways. That's glycolysis on this side and gluconeogenesis on this side. These are facts about the PFK1 that you've learned, and the critical thing is this activity that goes with gluconeogenesis is PFK2 and phosphofructokinase bisphosphatase are activities which are on the same protein. And so what happens that when this is phosphorylated, you change the activity. This is PFK2. It was named phosphofructokinase 2 because that's what the activity that was first found. People spent a lot of time looking for the other enzyme that would be the bisphosphatase before they realized it was the same. Notice in the major pathways for glycolysis where you have PFK1 and gluconeogenesis where you learn about fructose 1,6-bisphosphatase, these are two separate enzymes. For this regulation, we have a multiple domain protein that does the same thing. So when you have low cyclic AMP, because you're going to be putting all this in terms of cyclic AMP, it will favor the OH form, and this, PFK2, is an activator of glycolysis. If you have high cyclic AMP, you're going to phosphorylate that. And this is indicated by this cyclic AMP here. And so then you're going to lower the concentration of fructose 2,6-bisphosphate. So what you're going to do then is, and so when you do this, this destroys this and favors gluconeogenesis. If this fructose 2,6-bisphosphate is a stimulator of glycolysis, if you get rid of it, you're effectively inhibiting it. You're getting rid of something that stimulates, and that effect on the processes is going to inhibit it. Okay? So this is the inner circle. And they both, inner and outer circle, have the same substrate, fructose 6-phosphate. When we're on the pathway, we make fructose 1,6-bisphosphate and merrily go on our way because that's what we use for metabolism and other substrates. Metabolically speaking, as a substrate metabolite, fructose 2,6-bisphosphate is a dead end. The enzymes that will recognize fructose 1,6 don't recognize this at all. So this little inner circle going from fructose 6-phosphate to fructose 2,6-bisphosphate is essentially only involved in regulation. Okay? So it's a very important control mechanism. I alluded to, we didn't know about it before 1985, and you wouldn't have learned about it then. But because you've got this bifunctional enzyme, the active site is accessible in the OH form to act as a kinase. When it's phosphorylated, that site is closed up, and the phosphatase site is open. And the fact that cyclic AMP can do this, right away you should say, ah yeah, cyclic AMP, I learned about that in the first section, it's part of the hormonal systems. In the R section here, we'll go into much greater detail as to how these hormonal systems regulate metabolism, but a very important one is the cyclic AMP dependent systems, which have the hormone receptor isolated by epinephrine and glycogen. Okay, so this is, I'm introducing it here. All we've really had so far is this stage, going from fructose 6-phosphate to fructose 1,6, and it was just mentioned that fructose 2,6 is a stimulator. But when all these processes are studied, you're going to be responsible for the overall regulation. So think of this as a preview. And this slide will come back after we do gluconeogenesis and after we go and look at the glycogen system. So we see the interaction there with cyclic AMP. Okay, now oxygen and energy charge on glycolysis. This is just some information to show how we can monitor or we see what's going on. If you want to we have to maintain our steady state levels of our intermediate metabolites. And we can do this by turning glycolysis on and off. And we can measure this in anaerobic yeast cultures, because when they're growing on glucose, we can make them go aerobic, anaerobic, and you can see air off, you know, so you get these little bursts of air. And then when you do that, you wind up seeing that aerobic glycolysis is favored as it's much more efficient, and this is something we call the Pasteur effect. If you have a choice, why do fermentation that's only going to get you 2 ATP when you're going to be able to do oxidative phosphorylation and get 36? So when these studies were done, they also noticed this oscillation of the metabolites, sort of showing how you will change your concentrations of things important in energy charge, or the adenylate energy charge, as you turn glycolysis on and off. And that would make sense too, because as glycolysis is going on, that's sort of, hey, we're making energy, so let's not, we're going to be in good shape. And when glycolysis isn't happening, you have to be very parsimonious about what you make, because you have no former, sorry, because you have no guarantee that you're going to have enough energy to continue what you're doing. So when we look at metabolism being controlled in many different ways, we thought it can be called the distributive control. So we have enzymes in some pathways, not all, but in glycolysis we have a committed step and a major control step, and it's the same, phosphofructokinase 1. Quite often when you have committed steps and major control points, they're the same. Because from an efficiency point of view, why would you want to control the pathway beyond where you're committed to that pathway? Not all pathways have committed steps and major control points. Okay, and especially if it's not a linear pathway. So it's not, though, overall control is not simply a function of one enzyme, as you saw from the chart of glycolysis. That, in other words, you have allosterically regulated enzymes, feedback inhibition, etc. But when we talk about the regulation of the pathway, it's effectively the regulation of all the enzymes in those pathways summed up. But when you look at what's practical, any of the regulation at the committed step and the major control point is usually much stronger than the regulation you see for the other individual enzymes, and it also is usually acting in concert. Now, the critical thing about a pathway is that if you have a roadblock at any step, that's going to impede the flux through the entire pathway. So so when we're looking at these pathways, just like if you're missing one metabolite, you can't go on. If there's a blockage at any point, that blocks the whole pathway. And if you can't, like, say, well, I'll just use the first, if I have a block halfway through, you can't necessarily just use the first half of the pathway, because some of the controls for there came from later, some feedback, some substrate availability. Everything we talk about now, when we talk about delta G, and we say that under physiological conditions, the delta G is minus or positive, and when it's minus, we say it strongly favors this direction we want to go, and we call it spontaneous because it's essentially irreversible. I mean, under those conditions, if you're going to have to wait 10 years for a reaction to occur, you know, the metabolism boat has left. So essentially, from all practical purpose, that's irreversible. But this is tightly coupled to what the intracellular concentration of metabolites are. So if you block a pathway at any one point, you're going to get buildup of certain things, and you're going to have lower amounts of others, and you will no longer have what we call physiological conditions, also accumulating to what becomes the block of that pathway. Okay, so we're now headed to the second part of our anaerobic processes. We've done glycolysis, which is glucose breakdown, and now we're going to do glycogen breakdown, which is the polymer. So I'm going to go over the structure, the digestion, and mobilization, because we have to worry here about two different types of glycogen. You get glycogen in your diet, coming in through the GI system, and you store glycogen in the liver. Remember, when you think of a hotbed of metabolic activity, detoxification, it's all happening in the liver. So we also store most of our glycogen there, and we want to mobilize our internal glycogen. And that doesn't use the same pathways. Now, then we want to talk about the regulation of glycogen breakdown. And again, not surprisingly, there's going to be a phosphorylation, dephosphorylation involved. So you need to know the structure of glycogen, the role of glycogen phosphorylase, and the two functions of debranching enzyme in mobilization. You have to know when glucose and glucose 6-phosphates are released from glycogen, and how epinephrine and glucagon stimulate glycogen breakdown using adenylate cyclase. So it's the same cyclic AMP system that we talked about affecting the PFK2. So polysaccharide glycogens, they're polymers. They have large numbers of hexose units, and they have two different types of bonds, as you saw before, an alpha-1,4 and an alpha-1,6. Starch, potato, this is a polyglucose of plant origin. It's all alpha bonds. So how are we going to get rid of that? Amylose is a polyglucose with a linear chain, again, only alpha bonds. You're going to get that in. Amylopectin is a polyglucose with a branch structure, and it has both alpha-1,6 and alpha-1,4. So that means that when we're digesting this, we're going to have two different things, because what recognizes an alpha-1,4 is not going to recognize an alpha-1,6. Cellulose, that's a polyglucose with beta-1,4 bonds. We don't have cellulase, so we can't digest it. So cellulose, therefore, lacks caloric value. We can't burn it, but it does have nutritional value as fiber. So we have essentially pectins, ligand, and waxes, and it contributes to bulk and hydration, but you don't digest it. So when you take extra fiber, you'll be taking things that have cellulose with the beta-1,4. Glycogen is the polyglucose with the alpha bonds and highly branched structure, and it's the principal form of storage in the animals, and it's abundant mostly in the cytosol of liver, but we also see some in muscle, very little kidney and glial cells. Amylopectin, branch structure with alpha-1,6 and alpha-1,4, and glycogen have similar structures. So you could say, okay, I could have the same enzymes doing that. But you have to realize as we move on through this unit, we're talking about digestion. And if you think from the mouth to the anus, that's essentially outside the body. So just because we're going to put certain enzymes into the GI tract that are going to do that digestion, those enzymes certainly can't get across any cell membrane, and they're not available inside the cell to break down anything that the cell is making. So though the structure is similar, and you might be able to see similar specificities, the enzymes that are in the gut are not available in the cell, and vice versa. So you shouldn't be surprised that we're going to have different sets of enzymes. So here's the basic structure. We have your non-reducing end and your reducing end. And here you see in greater detail what an alpha-1,4 linkage looks like. And here's the alpha-1,6. So the OH here is the reducing end. You have one per molecule. But the non-reducing ends, as you branch, and you know this one will branch and this one will branch, so you'll have a lot of non-reducing ends per molecule, but only one reducing end. Okay, so when we break down that bond, well, there's two things we can do when we break down a glycosidic bond. We can hydrolyze it with a hydrolase, and therefore we essentially cleave it into two monomers, if this were a dimer, with OH groups there. So we can use this to some extent to polysaccharides in the gut. And then we can transport those polysaccharides across the plasma membrane by specific transporters to get them in so you can use them. But phosphorylase catalyzes this cleavage, and it also has the addition of a phosphate molecule. So you wind up with a free glucose and a glucose phosphate. This results in a phosphorylated sugar. It cannot leave the cell, and it enters glycolysis right away. So not surprisingly, you'd want to do this under conditions where you don't want glucose to get out of the cell. You don't want to do it under conditions in which you want glucose to get out of the cell to get to the brain. So what we're saying here, a kinase replaces the hydroxyl group with a phosphate group when it's phosphorylase, and a phosphatase will replace a phosphate group with a hydroxyl group. Okay, branch structure of glycogen. One of the advantages of it is it provides for compact storage. We store fat as glycogen, and we store excess energy as fat and glycogen, and the glycogen storage, if this were a long linear molecule, would be much bulkier than it is now, something that many of us are very grateful for. Now, it also, when we want to remember metabolism, we always want to do things quickly. We have multiple non-reducing ends here, or multiple ends that if we have an enzyme that's going to break it down from this end, if we had a linear form, we could only break one at a time. Here, we actually can start multiple enzymes at the time on the same molecule. So it has multiple sites to initiate the action of phosphorylase. Now, you know the two different types. Let's go through how we're going to do them, and polysaccharide digestion. So humans derive the glucose for energy from the two main sources: dietary starch, in other words, vegetable matter, and glycogen, because, well, if you're a carnivore, you're going to be eating animals that store this at various places. And they also do the mobilization. So the digestion, so when you're talking about digestion, that means in the gut. If you're talking about things from internal stores, we mobilize it. So starch and glycogen can be digested by the sequential action of several hydrolases. Some of them are in the mouth and some of them are in the gut. We have alpha amylase in the salivary glands in the mouth, and that will take care of alpha-1,4 glucosidase, and it's very pH sensitive. So for instance, if you have that enzyme that's secreted by the salivary glands, it's going to be inactivated in the stomach. You have pancreatic alpha amylase, which works on the same thing, cleaves off 2 units, maltose, and that one is active in the gut. We have a different enzyme, alpha-1,6-glycosidase. It's called the branching enzyme, and it's needed to act at the branch points because, you know, the alpha-1,4, you can just go taking them off, then suddenly you're at an alpha-1,6 linkage, can't do it, you need alpha-1,6. Then we have a maltase hydrolase, which works just on maltase, and lactase and sucrose. So this is a bunch of enzymes acting in concert. But when we do glycogen stores, that's a specific enzyme. So here's starch. This is a diagram. A lot of times I'll give you a picture and I'll give you words. Different people learn different ways. So they are saying the same thing. Here's your non-reducing end, your reducing ends, or in the alpha-1,6. So at the end of an amylase, where you're taking, in this case, off 2 units, you've got these alpha-1,6 hanging in the breeze, and alpha-1,6 glycosidase will break it down. Okay, now this is internal stores. And before we do the internal stores, because we're talking about cyclic AMP, I'll give you your break here for the moment. Okay. We're going to be constantly talking about hormones. This isn't one that's a yes or no again. So what we have, all through this course, we're going to be controlling metabolism, this section of the course, by regulation. And what you already know is that you've got a hormone receptor on the external surface of the cell that binds to your hormone. When we're talking about cyclic AMP, it's the G-coupled system is the receptor that's out there, and epinephrine and glucagon were the hormones. So the question is, can you think of a reason why the hormone receptor complex should ever wind up inside the cell? In other words, are these going to stay on the external surface, or should they be internalized? The whole point of this is the hormones are interacting with the receptor to basically transfer information. It's a signal that's saying, uh-oh, I have information out here that you should do this or you should do that. And the way of carrying it out is through the hormonal system. So just take a break and for a minute and think about this. I'm only going to give you a minute instead of 2 to try to make sure we get done before the 50-minute cutoff. No one wants to talk about yesterday's football game, huh? So quiet. Okay, so the way I ask the question, you should realize that it does happen. So does anyone have any thoughts as to why this would be a good idea? Yeah? Okay, well, in terms of metabolism, you have like 90% of stuff is the major pathways. When you talk about regulation, it's actually a small percentage of what's going in there. So these regulatory things are not likely to be detrimental. So that's not an obvious one. Mm-hmm. Yeah? You? Uh-huh. So therefore you want to, so why would you need to get it inside? Because the message is from the outside. You're moving in the right direction. Yeah. It's what? Making a tally. Okay, interesting idea. I don't think that's going on, but in terms of the tally, because what happens is the signal, when the minute the hormone hits the receptor, that's the signal. That starts whatever's going to happen, and then as far as information stored, we don't have to do what you're talking about with a tally. We've already figured that out. Okay. That's getting at the idea. Clearly, though, if there were more hormone, we want it. Oh, we have one more over here. Well, it depends on what you mean by faster. So let me go over what actually is happening and to see what's going on. All of the things that were mentioned are part of let's say, hormonal receptor metabolism. But what's going on is you have to do things quickly. So you need, from your point of view, if we had a lot of hormone, we'd want to have a lot of receptors out there because that hormone the formation of the hormone gets turned on and off. And if it keeps being on, we then want to make sure that signal is there. So therefore, we want empty receptors to be out there all the time. How are we going to get empty receptors? You do a recycling, because once the hormone and receptor bind, the information is done. We don't need it anymore. We can internalize it to degrade it, or sometimes just to separate out the hormone from the receptor and throw the receptor back out to be available for something else. So what we see it there for, it's a way of making sure there are always enough hormone receptors out there for the hormone, and a way of degrading the hormone, or taking the hormone off the receptor to allow that to happen. And again, we want to keep that cycling going quickly so we can keep our system going. Okay, so those were good ideas, all part of it a little bit, but not quite getting at the whole picture. But that's the important thing about these questions. You should think about it on the basis of your knowledge and come up with what you think is an idea, and then we'll build on it with everybody's different ideas when it's not a yes or no question. Okay, so we've done, before the question, we did the digestion. Now we're looking at mobilization of glycogen from internal stores. Phosphorylase is the major control point in this pathway. So that tells you we have a control point. And it's the enzyme that releases glucose from glycogen, cleaving the alpha-1,4 linkage. Okay? This enzyme works on our body glycogen, but it reaches that point at which you can't go any farther when you don't have the alpha-1,6. That sort of, from either case, digestion or internal, is called the limit dextran. And so phosphorylase, as it's working on alpha-1,4, releases glucose as glucose phosphate. The name kind of tells it to you. The digestion we did in the gut was not a phosphorylase, it was just a hydrolase. So those alpha-1,4s just released free glucose, and so that glucose could get into the cell. But now we're inside the cell. So we have an enzyme that releases glucose phosphate, and that glucose phosphate, as we saw in glycolysis, can't get out of the cell. If the body is sending a signal saying that we need more glucose, because that's why we're mobilizing our glycogen stores, then you certainly don't want whatever glucose that gets released to be sent out, unless we have another signal that the brain's in trouble. But in general, you want to keep it there. Debranching enzyme, so we've got a problem here. How are we going to get rid of it? And we have something new here, debranching enzyme. It's a bifunctional enzyme, and it first transfers 3 residues from a branch point, which is a transferase activity, missing an N there, and then releases the alpha-1,6 linkage. In the gut, we could use our less serious or less complex hydrolase to get right to the alpha-1,6 linkage. We can't do that here. In a typical glycogen molecule, there are 11 times more alpha-1,4 linkages than alpha-1,6 branch points. And since the debranching enzyme, when it hydrolyzes an alpha-1,6, it releases glucose as free glucose, this ratio here means we're going to have more glucose phosphates released in mobilizing from internal stores than free glucose. So an example of how this works. See, for some reason, phosphorylase can't get all the way in to free this alpha-1,6. So what you have, once you get rid of your alpha-1,4s, and this is what your dextran looks like, you then have the transferase activity, which will move 3 of these alpha-1,4s over here, giving you something like this. And then your alpha-1,4 can chop these off, and then these can get moved somewhere else, or something else can be moved there. But notice after this is moved, you now have an exposed alpha-1,6 linkage. And that one can be hydrolyzed as free glucose, and the debranching enzyme just will continue moving this to other forms. So it has to do with how phosphorylase not being able to get directly to the alpha-1,6. So we have muscle, which will use some glycogen as an immediate source of glucose, because if we're running a marathon or running from the saber-toothed tiger, we need to be able to get a little bit more energy, and that can be used locally. And other cells might contain small amounts, but the livers have the ability to pass glucose into the blood to deal with glucose homeostasis, and not surprisingly, liver will have the largest store of body glycogen, we'll see. Okay, so glycogen mobilization is hormonally controlled, and this is a diagram that, you may have even seen the similar one about the G protein. You've got your receptor here, epinephrine or glucagon, acting through your G protein, activating adenylate cyclase, the cyclic AMP protein kinase, And remember, when you activate this, all you can do is phosphorylate. So we have, in the OH form, phosphorylase B kinase, which is what does this, is inactive. At the action of cyclic AMP, we phosphorylase this enzyme, phosphorylase B kinase, and it's a two-step process because this is your first phosphorylation cascade. The bottom line is we're going to change from phosphorylase A, which is the phosphorylated form and active, to phosphorylase B, which is in the inactive form. And so first we have, this is the first one you've seen, of an enzyme called phosphorylase B kinase. Name tells it all. It's a kinase. It's putting on a phosphate for phosphorylase B. That's this enzyme's sole job. So it's important enough for the cell to actually have an enzyme who's totally involved in this regulation, and we call this a signal transduction cascade. And in this section of the course, only ones about glycolysis are you going to see. So our cyclic AMP is going to activate our regular protein kinase. This is fairly indiscriminate, and it will phosphorylate multiple things. Then it takes the inactive form of phosphorylase B kinase and phosphorylates it, and then the action of cyclic AMP through this will lead to the phosphorylation of phosphorylase B, turning it to phosphorylase A, and this will happen. Glycogen will be broken down. So this is a cascade that's an effective mechanism of single amplification, because that's what hormones do. They amplify signals. We have glucagon that would work here, and that's indicating raising cyclic AMP because you're hungry, or we have epinephrine, which is the stress hormone. So glucagon will work a little bit after meals or if you're starting a starvation. Epinephrine is stress. That's the saber-toothed tiger hormone. So they do have slightly different receptors. They're slightly different distributions in cells, but it is the G protein adenylate cyclase system for both of them. So activation of these kinases, this is just what I said, converts the inactive phosphorylase B into the active phosphorylase A. And that leads to the release of glucose 1-phosphate. Okay, so we finished Unit 2. And we can start Unit 3. So what we've done here is we've done our anaerobic processes. We've done glycolysis and glycogen breakdown. We've got our pyruvate sort of we sent it to pyruvate dehydrogenase. And now Unit 3 is oxidative processes. And we're just going to touch the surface of it here. The citric acid cycle, the glyoxylate shunt, and the pentose pathway. What these are all about are different carbohydrate utilizing systems that are oxidative processes. So the first set in this unit is citric acid cycle, Krebs cycle. So what we'll do is the overview of respiration and TCA cycle and discuss pyruvate dehydrogenase totally from a regulatory point of view as the link from pyruvate to the TCA cycle. Then in this pathway, I'll be illustrating by following specific atoms in the TCA cycle an interesting point that is true of most reactions but was first illustrated here. In the TCA cycle, we're going to put in acetyl-CoA, and that's 2 carbons, and 2 carbons are released as CO2. When this was first found, what was going on, the thought was 2 carbons in, 2 carbons out, they should be the same carbon. But what you'll see from the enzyme mechanisms that we're not doing in great detail here, that for each, when a molecule of acetyl-CoA is put into the TCA cycle, those 2 carbons are not the 2 carbons that come out in the first round of the cycle. We'll see this here. That idea exists all through metabolism. This is only 20 lectures, so therefore I'm only illustrating it here and not pointing it out in other places where it's true. And why this happens then becomes an interesting point. And from and what the basic answer is, enzymes can treat what chemically are considered symmetrical molecules asymmetrically. Because when you look at it from a chemical point of view, you're looking at a molecule all by itself. An enzyme has a plane and an active site embedded in that plane, and therefore you can introduce some symmetry there. Then we'll do the energetics of the cycle. At the end of this, we will have oxidize our glucose to CO2 and reduce nucleotides. And then we have this pathway there. The cell does use it for other things, and that's obtaining metabolic intermediates, and that's called anaplerotic pathways, filling up pathways. That's what you do when you want to do biosynthesis. So what are the important points coming up here? So you have to understand how pyruvate dehydrogenase works to regulate metabolism, because once you get to acetyl-CoA, you can't use it to make anything else. So it's a major control in intermediate metabolism. It's controlling what's available to enter the TCA cycle, because the TCA cycle has no major control point. And you have to know how it generates the intermediates that can replace the chemicals that need to be. So it has two functions. If you need to use it to make something, you can feed in its a cycle somewhere and pull off somewhere else. But if you use it to make energy and just run it around, that's being used catalytically. And so when you do that in energy metabolism, it's a catalytic use. So this cellular respiration process occurs aerobically inside the mitochondria. And again, the commitment to this aerobic respiration occurs at the pyruvate dehydrogenase step. This could be considered the activation step for the TCA cycle, even though it's not part of the cycle, and that's a metabolic principle. Now, commitment to TCA, the enzyme that's doing this, is the citrate synthase reaction that's making citrate. So in the TCA pathway, you need to know which steps generate NADH, which steps generate an energetic ATP equivalent, which is GTP. I'm focusing on energetics in this section. We're talking about making ATP and oxidative phosphorylation. But when we look at a net energy balance, a cell can trade an ATP to a GTP. You know, an ATP can transfer that terminal phosphate to GDP to form GTP, and that is no energy change. It can do this without losing or gaining energy. So I think of making a GTP as energetically equivalent to making an ATP, and we will do this at one point in the TCA cycle. That's the substrate level phosphorylation that occurs in the TCA cycle. So you have to be able to follow each carbon atom and know why it takes more than one cycle to burn the exact two carbons that enter this pathway. If you look through the problem sets in both exams, there's a question involving in the energetics where I ask you the consequences of going from one place in the TCA cycle to another place in the TCA cycle. And in order to answer that question, you have to know the TCA cycle pretty well to know where the GTP is made and where the NADHs are made. So again, how this asymmetry occurs, you need to recognize the symmetrical citrate effects of the TCA cycle because the enzyme can consider it to be not symmetrical. So let's figure out where this respiration is in the overall scheme of things. It's our stage 3 metabolism. So we've done one example of stage 1, which is glycogen going to monomers, monomer being glucose. And we had glycolysis as our stage 2. And then we get to respiration, which is our stage 3 in metabolism. And unfortunately, I don't like that they do this, but they do. They then divide this respiration into 3 steps. So it's part of stage 3 metabolism, and it itself has 3 stages. The first one is what we're doing up here when we're essentially doing pyruvate dehydrogenase. The second stage is the TCA cycle, which is this unit. And the third stage is oxidative phosphorylation, which is unit 4. So our respiratory processes that we're into are complex. They have stages of their own, and together they make up what we call stage 3 of metabolism. Whoops. Okay. So these are the 3 parts, and this is the words before was the picture, and the picture's now blown up. It's the commitment to aerobic oxidation. It's the activation of intermediates. It's a family of pathways, because pyruvate can be used to do other things. But once you do acetyl-CoA, and we can get acetyl-CoA in other ways, not just from pyruvate dehydrogenase, from amino acids and fatty acids, but this is a 2-carbon. And we'll see from this, if we're going to use the TCA cycle, 2 carbons in, 2 carbons out, just using this version of the intermediate point at acetyl-CoA, we can't make anything. There can be no net synthesis of carbon, of amino acids or anything you want to make, if 2 carbons go in and 2 carbons go out. So that's why this is the commitment to the aerobic oxidation of breaking things down, because it's very difficult. We can do it through using parts of the TCA cycle, the anapleuratic reactions. We can then make glucose if we want, but it's going to be at a high energy cost. So this is the generation of the CO2 and our reduced nucleotides, NADH and FADH, which are going to happen in the TCA cycle. Again, that's through this unit. And stage 3, we have to regenerate. We're always having to regenerate what we use, our electron carriers. And to do that, we get the generation of water from oxygen and phosphorylation, finally, of ADP to ATP. So as I said earlier, at the end of this, we will have broken it down to our CO2 and reduced nucleotides. After our reduced nucleotides go through the respiratory chain, we will have made our ATP, and we will have broken it down to water and made our ATP. So that's the overview through section 5 here, and we're in unit 5, and we're in the middle of unit 3. So I always like to point out to you guys that this is like the real world, and so where do we get the energy for cellular respiration? We get it from breathing. Okay, there's no special mechanism going on. Now, for those of you who are studying physiology, well, whenever you study, whether you're taking it now or at some other time, the details of how the oxygen gets into the cells that you breathe will go over it. So this is, when we call respiration, human respirations, it's the use of oxygen. And so we also have the production of CO2 or heat by the organism. So these quantities, changes in CO2, oxygen, and heat, all reflect metabolic activity. They're not all proportional to each other. So this is the respiration that the physiologists talk about. You know, and when you breathe hard, it's that. Cellular respiration processes are how that oxygen gets utilized inside the cell. And it's a process in which the chemical energy that we have stored in our bonds of our fuels, and remember, fuels are just like what you talk about in nutrition, carbs, lipids, and proteins. They're converted into energy that we need for life processes, which is ATP, and that's the internal combustion that's happening, and it requires oxygen. It's the cellular respiration process does. Remember, glycolysis didn't, but cellular respiration does, and it generates CO2 and water. So in its slow way, we do eventually get our CO2 and water, similar to if we actually burned our fuels, but in the process, we're able to get out a lot more energy. So breathing, I can't pronounce it, is concerned with the transport of the oxygen from the air to tissues. And once you get into the tissues, cellular respiration takes over. And what we have to worry about, or what the physiologists then worry about, I don't, in metabolism, is getting rid of that CO2 that we generate. Obviously, it's not going to stay there. And so in air and gas, we have 78% nitrogen and 21% oxygen here, and various amounts of water vapor. Okay, so I just want to place the TCA cycle here in the center. Pyruvate to the acetyl-CoA, we're going to get to this cycle, and that's what we're going to be explaining, and I believe we start with the overview, and that's about as far as I want to go today, and I think I'm all within it. When I get back from my trip, I'm going to see what I can do about this new system, but for the moment, I think you have to count on being cut off at 9:50, and I'll try very hard to stay to it.